mode and
Great.
everything. Thanks, sir.
Thank you, Dr. Berry, and thank you to the Symposium for inviting us to present today on M102, an NRF2 and HSF1 activator entering phase 1 for the treatment of ALS. We are at the tip of the iceberg in developing powerful and transformative gene therapies that will change the lives of many ALS patients. However, the vast majority of our patients will remain in need of treatments. ALS is a diverse and multifactorial disease, and current treatments do not account for disease heterogeneity and the multiple mechanisms that drive neural injury. Key pathways include redox balance, inflammation, mitochondrial dysfunction, and altered proteostasis. I'm here to discuss NRF2 and HSF1, two targets that we at Aclipse believe are relevant to and potentially druggable in a large percentage of ALS patients. Some of the earliest evidence for dysregulation of NRF2 pathway signaling were generated in cellular models of ALS in Dame Pamela Shaw's lab. Pam's lab actually discovered M102, and much of the data I showed today was generated in the lab. Pam's group at Citran used the mouse SOD1 gene 93A transfected motor neuronal cell line, NSC34. They showed a broad general transcriptional repression of both NRF2 itself and NRF2 target genes, as indicated in the diagram on the right-hand side. The reduced activation of NRF2 was shown to be corrected by pharmacological NRF2 pathway activation. Small molecule electrophiles were able to overcome this repression, seemed to be seemed to be restricted to mutant SOD1 transgenic specific models. Using human ALS tissue, Pam and her team were able to use a proteomic approach to further show that PRDX3, a key NRF2 regulated mitochondrial antioxidant enzyme, was downregulated in both the cellular model of ALS and in motor neurons. In the lower figures, motor neurons from healthy control in ALS patients were stained for PDRX3, which is believed to lead to reduced protection of mitochondria during oxidative stress. This was also corrected by NRF2 pathway activation. Further evidence of decreased NRF2 expression in ALS was presented by examination of sporadic ALS postmortem samples. Motor cortex and spinal cord motor neurons in 5 ALS patients and 5 matched controls were evaluated. X-ray autoradiograms of NRF2 mRNA expression in the primary motor cortex of ALS patients and controls are seen in the first row, with separation across cortical layers 1 to 2, 3 to 5, and 6. mRNA expression was assessed semi-quantitatively by densitometry of the film autoradiograms. Additional studies illustrating optical densities representing ratio to beta-actin on Western blots were performed, and these studies similarly demonstrated diminished NRF2 expression in sporadic ALS. Dr. Chen at UCL noted the presence of reduced HSF1 and heat shock proteins in both the transgenic mouse model and in spinal cord tissue from sporadic ALS patients. She proposed that compromised HSF-mediated HSP70 response was a contributory pathway in ALS, and she designed an elegant experiment to study this. During cellular stress, HSF1 dissociates from HSP90, trimerizes, and translocates into the nucleus where it binds to the heat shock element through its DNA binding protein. There, it promotes the transcription of downstream genes via its transactive domain. Using HEC293 cells that were transfected to overexpress wild type or 2 different mutant TDP43 types, she co-infected with either a dominant active HSF1 whose regulatory domain was deleted and could not be inactivated by HSP binding, or alternatively, a dominant inactive construct whose transactive domain was deleted. Expression of dominant active HSF1 resulted in a dramatic reduction of insoluble TDP43 that enhanced cell survival in both TDP mutants, whereas expression of dominant negative HSF1 led to enhanced TDP43 aggregation. Levels of soluble TDP43 were only marginally affected. M102 is a potential disease-modifying drug for ALS. Designed to affect multiple pathomechanisms, M102 is the only NRF2 and HSF1 activator. Multiple data sets in numerous animal models, some of which were presented yesterday by Dame Pamela Shaw, support the ability to halt disease progression, revert TDP-43 proteinopathy, and extend motor neuron survival. M102 is the S enantiomer of apokine, a known Parkinson's drug. It crosses the blood-brain barrier and is being developed as an oral agent. We have a patient stratification strategy through a proprietary gene map we were developing that potentially will identify drug responders as well as enrich the clinical study patient population. Composition of matter extends to 2040, and we have completed a pre-IND meeting with FDA and intend to start our SAD/MAD study in the first quarter of next year. M102 is the mirror image of apokine, but as noted, has significant differences. Chief among them is that it is a new chemical entity, a strong NRF2 and HSF1 activator, a weak dopamine antagonist versus a strong dopamine agonist, and not substitutable. Importantly, the apokine safety and CMC database de-risk the M102 program. This is a simplified schematic that demonstrates the two biological targets for M102. Both NRF2 and HSF1 are inactive in the cytoplasm due to their association with their regulatory partners. NRF2 is bound to KEAP1, which interacts with NR2 and leads to its sequestration and subsequent degradation by the ubiquitin proteasome system. In the presence of oxidative stress, M102 is activated to its orthoquinone active moiety, which is able to deubiquinate NRF2 and free it to translocate into the nucleus, where it can upregulate greater than 250 genes, including glutathione and others implicated in ALS. Similarly, HSP90 is bound to HSF1 within the cytoplasm and negatively regulates it. M102 is able to liberate HSF1 and allow it to enter the nucleus and upregulate expression of heat shock proteins that form the basis of the chaperone system and facilitate disaggregation of TDP-43. As noted yesterday, there are many pathological pathways implicated in ALS, and we believe an optimal approach is to target those broadly with strong NRF2 and HSF1 activation. As you see, currently approved therapies are limited in their approach. This data demonstrates statistically significant NRF2 and HSF1 target engagement in wild-type mice. The gene expression with transcriptional activation of wild-type mice spinal cord is expressed 24 hours after completion of 7 days of once-daily subcutaneous M102. As you see, there's a nice separation from vehicle control in both the NRF2 and the HSF1 targets. Turning towards an ALS model, this is a depiction of target engagement in TDP-43 Q331K mice. These animals were dosed subcutaneously to 3 months at either 2.5 milligrams BID or 5 milligrams once a day, and demonstrated NRF2 and HSF1 target engagement. Just pointing out that HSP8 is synonymous with HSP70. Glutathione is an important cellular regulator, and reduced glutathione scavenges free radicals to reduce oxidative stress. As such, glutathione is a potential biomarker for oxidative stress, and importantly, can be measured across species. Dysregulation of glutathione homeostasis is believed to contribute to ALS progression, as reduction in glutathione and glutathione oxidized glutathione ratio has been observed in ALS patients. One of the things that we are excited about is the increase in glutathione and glutathione ratio seen with M102 in mouse cortex in the wild-type mouse experiment. Of note is the apparent lack of increased benefit with higher dose, but this will be evaluated further in our development program. Personally, I'm actually quite I'm quite excited and looking forward to glutathione data apart from ALS. The very first experiments and research I did as a pre-med student entailed assessing rat liver homogenates for glutathione activity. So it's nice to actually come back to the molecule. Pam reviewed this yesterday, so I won't spend a great deal of time on it. Essentially, using ALS patient fibroblasts, one may develop induced astrocytes and motor neurons that retain the genetic and phenotypic profile of the original donor ALS patient. Given that astrocytes control motor neuron survival, we use this model in some of the data I will be sharing. Using R, a computer programming language, differentially expressed genes after M102 treatment were identified in control and patient eye astrocytes, and we're analyzing using data analysis software to identify pathways significantly altered in each donor subgroup after treatment. There is a broad, statistically significant response in ALS patients across a patient spectrum, C9, SOD1, and sporadic ALS patients. Interestingly, M102 elicits a differential gene expression response in patients and controls, probably due to its activation in conditions of oxidative stress. Pam showed this data yesterday, but it merits repeating. TDP-43 accelerates cytoplasmic mislocalization and accumulation of TDP-3 toxic aggregates. TDP-43 dysfunction is present in 97% of ALS patients, and many believe that TDP-35 is what drives the neurotoxicity in ALS. A therapeutic that might be able to reverse this or lessen it would be highly beneficial to patients. 10 micromolar concentration M102 was added to patient-derived induced astrocytes. There were 10 sporadic ALS patient samples and 3 C9 ALS patient samples. Measurement of both TDP-43 and TDP-35 was conducted post-drug administration. A control is shown to confirm the normal absence of TDP-35. We see a time-dependent reduction in TDP-43 proteinopathy and a shift in the TDP-35-TDP-43 ratio. Of note, this took 24 hours in sporadic ALS and longer in C9 ALS. CMAP, or compound muscle action potential, is a measure of muscle health and is also translatable across species. It consists of stimulating a nerve fiber and monitoring the response in the corresponding muscle. We evaluated CMAP in the TDP-43 mouse model in an effort to see what effect 5 months treatment with M102 would have versus vehicle. As expected, CMAP decreased by 20% in the untreated mice, while M102 treated mice exhibited a dose response of 50% improvement versus untreated animals. This is greater than that of any FDA-approved drug and suggests that M102 could be a disease-modifying therapy that reverses disease progression. We also looked at M102's ability to rescue SOD1 transgenic spinal motor neurons and were successful in increasing motor neuron survival within lumbar ventral horns when compared to vehicle-treated animals at 90 days. Dose-dependent improvement in survival approached that seen in healthy wild-type mice. Given the multiplicity of disease genotypes and phenotypes, it behooves all of us to develop targeted or precision therapies that provide effective treatment to those best suited to respond. This is not only the case for ALS, but a major focus in oncology and other conditions. Our goal is to develop a broad-based dual NRF2-HS1 activator that will allow identification of and enrichment for potential M102 responders. Using co-cultures of induced astrocytes and mouse motor neurons, we examined astrocyte RNA-seq data pre- and post-treatment. We identified potential strong responders that exhibited improved motor neuron survival, as well as weak responders with poor motor neuron survival. Upon treatment with M102, the strong responders' heat map was demonstrated to revert to that of healthy controls, while poor responders showed no change. We hope to develop a PBMC assay that will allow identification of strong versus weak responders on the basis of a 3 to 4 gene panel. One cannot discuss an upcoming phase 1 program without preclinical toxicology. We conducted GLP 28-day tox studies in 2 species, rats and non-human primates, dosing at 25, 50, and 75 milligrams for 4 weeks with a 2-week recovery period. M102 was well tolerated with no adverse findings noted. NOEL was determined to be 75 milligrams per kilogram, and using in vivo efficacy and calculations adjusting for the difference in free fraction of drug, we expect an 18 to 27 times TOC margin for the expected efficacious human dose. We're gearing up for first-in-human studies early next year. Our SAD/MAD study will enroll 80 healthy volunteers in a cohort of ALS patients. We will establish the safety profile of M102 through supratherapeutic exposures and assess PK/PD in plasma as well as PK/PD in CSF and move over to the MAD. Target engagement will be evaluated via PMVCs as well as upregulation of NRF2 and HSF-CSF. In the ALS cohort, we will dose through 28 days to gain a better assessment of target engagement. Based on safety monitoring committee recommendation and demonstration of pharmacodynamic effect, may initiate the MAD while still progressing through the SAD cohorts. This is the final bit of data I will share today, and it's important because it shows the potential benefit over currently approved small molecule drugs for ALS. Studying induced motor neurons from ALS patients, M102 successfully rescued motor neurons in 7 of 9 SOD1, C9, and sporadic ALS patients, outperforming riluzole, edaravone, and the active agent Tecfidera, which is approved in relapsing remitting MS. In summary, M102 is the first dual NRF2 and HSF activator designed to attenuate the major pathomechanisms present in ALS. Extensive in vivo and in vitro experiments across ALS subtypes support the disease-modifying potential of M102. We're pursuing a genetic biomarker panel to enrich our patient population for strong responders. M102 is an oral drug with composition of matter protection until 2040 and plans for our phase 1 clinical trial notification early next year. I'd like to thank Dr. Berry and the organizers of this symposium. I'd also like to thank both US-based colleagues at Aclipse, as well as our wonderful colleagues across the pond, as well as our collaborators. And finally, thank you to the organizations that provide the funding to help small companies and academic researchers advance their dreams of making the world a better place, one drug at a time. Thank you.
Really wonderful presentation. Thank you so much. We do have a few questions, and we have just a couple of minutes. One question is actually about a comparison to another drug. As you said, M102 acts as a redox regulator. In slide 17, why don't you use edaravone comparing M102? Why did you use riluzole?
So we actually are in the process of doing that.
Okay, interesting. Another question is about safety. Could you comment on the activator of HSF1 and the potential for oncogenesis?
So we think it generally will only be seen in oxidative stress. But certainly there is a possibility for oncogenesis. We've conducted a series of preclinical testing that basically has supported our ability to go straight into humans. We've done a big blue study, pig studies, and we've done the necessary testing that we feel is going to allow us to go into the clinic. And it's our expectation, the expectation of the regulators that we've spoken to, that we have a molecule that will be safe. Again, this is an ALS patient population. If there is a risk, it may take 20 years or 30 years for that to come across. We're pretty confident that we'll be able to do something for the disease and able to minimize any issues going forward on the oncocyte.
There's a question: is this activation also able to overcome the high threshold in neurons, or just amplify the competent response in astrocytes? And this person comments that they may have missed if there were also iPSC-derived neurons tested.
So we are basically using iNPC neurons tested. We are in the process of repeating the experiments using motor neurons itself.
Okay. And then the last question, and then we have to move on, but there are more in the chat. Sorry, I can't get to all of them. But since M102 binding is covalent, do you expect this effect to be irreversible?
I think we haven't studied. I mean, we know that when we look at PK, we see what levels are done. The body is constantly recreating. So, do I think it's going to be a problem? We don't expect to see basically accumulation. So when we talk about irreversibility, it comes down to a really cellular level, and without accumulation, I don't think it's something that we're going to be having to deal with.
Okay, thank you so much. Wonderful presentation, really exciting science. Thank you for
Thank you.
Sharing with us.